After we have clustered the cells and visualized them with dsNE or UMAP, it is time to find conserved biomarkers for the clusters and look for differentially expressed genes between the samples or sample groups within the clusters. And finally, visualize some interesting genes. In Chipster, we offer a couple of tools for these steps, which you can do one cluster at a time. The visualization can be done to several genes at a time. When talking about conserved marker genes, we mean markers for a given cluster in all the samples. In this tool, you give a cluster as a parameter, and the tool compares the gene expression in that cluster versus all the other cells in the data. This is done for each sample of the data, and then the p-values are combined. This test uses Wilcoxon-rank sum method. The conserved markers result table looks like this. So you will have the statistical values for both groups. Here we have control and stimulated. And then the combined maximum p-value and minimum p-value, which you can use to filter the table. For finding the cell type specific differentially expressed genes, we have a couple of tools. You can choose to do this to all the samples in the data, or you can choose some samples or perform it to sample groups. Now, in the case where we have only 2 samples, all the tests will give the same result. But when you have multiple samples, this of course makes a difference. So what we are looking now is the differential expression between specified samples or sample groups, again in one cluster. The result table looks very similar to the conserved markers table, and you can filter it in the same way. If there are more than 2 samples, a table for each sample is given as an output, and they are named according to the sample names. Now, when giving the sample or sample group name to the tool as a parameter, you might want to check from the setup tool which parameters you used there. So for the sample group, it would be this parameter, and for the sample, this name here. Step in this 2-sample pipeline is the visualization of interesting genes. These genes can come from the list of conserved markers, or they can be the differentially expressed genes between the samples, or just your personal genes of interest. This plot, that we like to call the Marimekko plot, shows 2 things at once. The percentage of cells expressing a gene is shown as the size of the dot, and the brightness of the dot represents the expression level. On the x-axis, we have the genes that we chose for the plotting, and on the y-axis, we can see the different clusters separated also based on the sample. The sample is also highlighted with the color. Blue here is for the control sample and red for the stimulated sample. Here you can see, for example, that these 2 genes seem to be excellent conserved cluster markers for cluster number 12. Here we see some genes that seem to behave differently in the different samples. We can also use the good old dsNE and UMAP plots to see the expression of the selected genes in different scenarios. In scenario number 1 here, there's very little difference between the samples. These 2 genes are thus conserved cell type markers. In scenario number 2, there's very little expression in control sample and lots of expression in the treated sample. So these 2 genes here are cell type independent markers for the treatment. In the last scenario, we see cell type dependent reaction to the treatment. This gene is active in this cluster here in the control sample, but not in the treated sample, and then vice versa with this gene. We can see the same 3 scenarios here in the violin plots. Here on the x-axis, we see the different clusters, and for each of these 3 genes, we see the expression level in control, red, and treated, green. Conserved markers show somewhat similar expression in both samples. This gene here is affected by the treatment regardless of the cell type, so we see strong difference between the samples. In the last plot, we can see that some clusters or cell types are reacting to the treatment and others are not.